11th class, NEET students, audiobook, Chapter 6, Biology, Anatomy of Flowering Plant. You can very easily see the structural similarities and variation in the external morphology of the larger living organism, both plants and animals. Similarly, if we were to study the internal structure, one also finds several similarities as well as differences. This chapter introduces you to the internal structural and functional organization of higher plant. Study of internal structure of plant is called anatomy. Plants have cells as the basic unit. Cells are organized into tissues, and in turn the tissues are organized into organs. Different organs in a plant show differences in their internal structure. Within angiosperm, the monocote and the dicote are also seen to be anatomically different. Internal structure also show adaptations to diverse environments. The tissue system. We were discussing types of tissue based on the types of cells present. Let us now consider how tissues vary depending on their location in the plant body. Their structure and function would also be dependent on the location. On the basis of their structure and location, there are three types of tissue system. These are the epidermal tissue system, the ground or the fundamental tissue system, and the vascular or conducting tissue system. Epidermal tissue system. The epidermal tissue system forms the outermost covering of the whole plant body and comprises epidermal cell, stomata, and the epidermal appendages, the trichomes and hairs. The epidermis is the outermost layer of the primary plant body. It is made up of elongated, compactly arranged cells which form a continuous layer. Epidermis is usually single layered. Epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell wall and a large vacuole. The outside of the epidermis is often covered with a waxy thick layer called the cuticle, which prevents the loss of water. Cuticle is absent in roots. Stomata are the structures present in the epidermis of leaves. Stomata regulate the process of transpiration and gaseous exchange. Each stomata is composed of two bean-shaped cells known as guard cell, which enclose the stomatal pore. In grasses, the guard cells are dumbbell-shaped. The outer wall of the guard cells away from the stomatal pore are thin, and the inner wall towards the stomatal pore are highly thickened. The guard cell possesses chloroplast and regulate the opening and closing of stomata. Sometimes, a few epidermal cells in the vicinity of the guard cells become specialized in their shape and size, and they are known as the subsidiary cells. The stomatal aperture, guard cells, and the surrounding subsidiary cells are together called stomatal apparatus. As you can see in the figure 6.1 diagrammatic representation, A figure represents the stomata with open bean-shaped guard cell, and B represents the stomata with dumbbell-shaped guard cell. A is for the dicote plant and B is for the monocote plant. At the outer side there is epidermal cells, and there is modification of some epidermal cells into the subsidiary cells which are present in the vicinity of the guard cell. Around the guard cell there are subsidiary cells which are shown in the dark green color here. In the light green color there is guard cell into the middle, and there is a stomatal pore which is white in color inner to the guard cell. Inside the guard cells there are some dotted structure which are representing the chloroplast, and this is the area which is useful for gaseous exchange and for the water transpiration. In the dicote you can see the guard cells are of the bean shape, and in monocote the guard cells are of the dumbbell shape. Now the cells of the epidermis bears a number of hairs. The root hairs are unicellular elongation of the epidermal cells and help absorb water and minerals from the soil. On the stem, the epidermal hairs are called trichomes. The trichomes in the shoot system are usually multicellular. They may be branched or unbranched and soft or stiff. They may even be secretory. The trichomes help in preventing water loss due to transpiration. The ground system. All tissues except epidermis and vascular bundles constitute the ground tissue. It consists of some simple tissues such as parenchyma, collenchyma, and sclerenchyma. Parenchymatous cells are usually present in cortex, pericycle, pith, and medullary rays, in the primary stem and in the roots. In leaves, the ground tissue consists of thin-walled chloroplast containing cells and is called the mesophyll. The vascular tissue system. The vascular system consists of complex tissue such as the phloem and the xylem. The system and phloem together constitute vascular bundles. In dicotyledon stem, cambium is present between phloem and xylem. Such vascular bundle, because of the presence of cambium, possess the ability to form the secondary xylem and phloem tissue and hence are called open vascular bundle. In the monocotyledons, the vascular bundles have no cambium present in them. Hence, since they do not form the secondary tissues, they are referred to as closed. When xylem and phloem within a vascular bundles are arranged in an alternate manner along the different radii, the arrangement is called as the radials, such as in roots. In conjoint type of vascular bundles, the xylem and the phloem are jointly situated along the same radius of the vascular bundle. Such vascular bundles are common in stem and leaves. The conjoint vascular bundle usually have the phloem located on the outer side of xylem. In the figure, you can see various type of vascular bundle. A figure represent the radial vascular bundle, B figure represent the conjoint closed vascular bundle, C figure represent conjoint open vascular bundle. In A figure, you can see the xylem and the phloem are present on different radius. They are not along with each other. That's why this is called as a radial type of vascular bundle. In second figure, in B figure, phloem and xylem both are present along the same radii in the same radius, but they do not have the cambium present in between phloem and xylem. That's why it is not going to show secondary growth and it is present into the monocotyledons. There is no secondary growth and this condition is referred as conjoint closed. In the C figure, you can see there is a presence of cambium between the phloem and the xylem and it represents the conjoint open condition where secondary growth is possible and it is present only in the dicotyledon plant. Anatomy of dicotyledons and monocotyledon plant. For a better understanding of tissue organization of roots, stem and leaves, it is convenient to study the transverse section of the mature zone of these regions or these organs. Dicotyledon root. Look at figure 6.3a. It shows the transverse section of the sunflower root. As it is a sunflower, that's why we can say it is a dicot root. The internal tissue organization is as follows. First of all, in the dicotyledon root, the outermost layer is epiblema. Many of the cells of epiblema protrude in the form of unicellular root hair. The cortex consists of several layers of thin-walled parenchyma cell with intercellular spaces. The innermost layer of the cortex is called the endodermis. It comprises a single layer of barrel-shaped cells without any intercellular spaces. The tangential as well as the radial wall of the endodermal cell have a deposition of water impermeable waxy material suberin in the form of Casparian strip. Next to endodermis lies a few layers of thick-walled parenchymatous cells referred to as pericycle. Initiation of lateral roots and vascular cambium during the secondary growth take place in these cells. The pith is small or inconspicuous. The parenchymatous cells which lie between the xylem and the phloem are called conjunctive tissue. There are usually 2 to 4 xylem and phloem patches. Later, a cambium ring develops between xylem and phloem. All tissue on the inner side of the endodermis such as pericycle, vascular bundle and pith constitute the stele. In figure A, you can see the outer layer is epidermis, which is shown in pink color, and from the epidermis there are protrusions which is called as root hair, and in the green layer you can see there are multiple layers of parenchymatous cells which form the cortex. The inner layer of the cortex is known as endodermis, and inside the endodermis there is a pericycle which is made up of barrel shaped cell, and there is a presence of vascular bundle. 2 to 4 xylem and phloem patches are present. There is a protoxylem and metaxylem. Protoxylem are present towards the pith, and protoxylem are present towards the pericycle, and metaxylem are present towards the pith. This condition is called as exarch condition. Pith is present into the center, and phloem is present between the xylem. That's why radial type of vascular bundles are present. Monocotyledon root. Figure 6.3b figure represent monocotyledon root. The anatomy of the monocot root is similar to the dicot root in many aspects. It has epidermis, cortex, endodermis, pericycle, vascular bundle, and pith. As compared to the dicot root, which have fewer xylem bundle, there are usually more than 6 polyarch xylem bundles in the monocot root. Pith is large and well developed. Monocotyledon's roots do not undergo any secondary growth. Remember, monocotyledon's roots do not undergo any secondary growth because there is no presence of cambium, and only conjoined closed vascular bundles are present. Only radial and closed vascular bundles are present because cambium is not present. Again, exarch conditions are present, and the similarity is exactly similar to the dicotyledon root. Dicotyledon stem. The transverse sections of a typical young dicotyledon stem shows that the epidermis is the outermost protective layer of the stem, covered with a thin layer of cuticle. It may bear trichomes and a few stomata. The cells arranged in a multiple layers between epidermis and pericycle constitute the cortex. It consists of 3 subzones. The outer hypodermis consists of few layers of sclerenchymatous cells just below the epidermis, which provide mechanical strength to the young stem. Cortical layers below hypodermis consist of rounded thin wall parenchymatous cells with conspicuous intercellular spaces. The innermost layer of the cortex is called the endodermis. The cells of the endodermis are rich in starch grain, and layer is also referred to as the starch sheath. Pericycle is present on the inner side of endodermis and above the phloem in the form of semi-lunar patches of sclerenchyma. In between the vascular bundle, there are few layers of radially placed parenchymatous cells which constitute the medullary race. A large number of vascular bundles are arranged in a ring. The ring arrangement of the vascular bundle is a characteristic of the dicot stem. Each vascular bundle is conjoined, open, and with endark protoxylem. A large number of rounded parenchymatous cells with large intercellular spaces which occupy the central portion of the stem constitute the pith. Monocotyledon stem. The monocot stem has a sclerenchymatous hypodermis, a large number of scattered vascular bundle, each surrounded by a sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. Vascular bundles are conjoined and closed. Peripheral vascular bundles are generally smaller than the centrally located one. The phloem parenchyma is absent, and water-containing cavities are present within the vascular bundle. In figure 6.4, transverse section of stem, A figure represent the dicot stem and B figure represent monocot stem. In A figure, you can see there is outermost layer which is epidermis, and from the epidermis, epidermal layers are arises. Below the epidermis, there is a portion of cortex, and the upper portion of cortex is formed hypodermis. In dicotyledon plant, hypodermis stem in dicot plant, stem hypodermis made up of collenchyma and parenchyma. Then below the hypodermis, there is a cortical cells which are mainly form of parenchyma. The innermost zone of the cortex form the endodermis. Below the endodermis, there is pericycle, And below the pericycle there is vascular bundle, that is phloem and xylem. Between the phloem and the xylem there is presence of cambium, which represent the secondary growth formation. And the metaxylem is present towards the phloem and protoxylem towards the pith. That condition represent the endark condition of vascular bundle. Now between the vascular bundle there are presence of laterally attached, laterally formed parenchymatous cells bundle, which constitute medullary rays which help in the lateral conduction of food and water. In the center there is a large pith. So in white color pith is shown, which is very large. But with compared to monocot, in the monocot stem the outermost layer is epidermis, and below the epidermis there is hypodermis. Hypodermis is made up of sclerenchyma, and the vascular bundles are present. There is not a well differentiated ground tissue, and vascular bundles are scattered everywhere. In the center there are large size vascular bundle, and in the periphery there are small sized vascular bundle, and in between the ground tissue is completely scattered or completely diffused. Phloem and xylem are present as vascular bundle. There is no cambium, so no secondary growth. And conjoint or closed type of vascular bundles are present, whereas in dicotyledon plant conjoint and open type of vascular bundles are present. Dorsiventral leaf, dicotyledon leaf. The vertical section of a dorsiventral leaf through the lamina shows the three main parts, namely epidermis, mesophyll and vascular system. The epidermis, which covers both the upper surface adaxial epidermis and the lower surface abaxial epidermis of the leaf, has a conspicuous cuticle. The abaxial epidermis generally bears more stomata than the adaxial epidermis. The latter may even lack stomata. The tissue between the upper and the lower epidermis is called the mesophyll. Mesophyll, which possess chloroplast and carry out photosynthesis, is made up of parenchyma. It has two types of cell, the palisade parenchyma and the spongy parenchyma. The adaxially placed palisade parenchyma is made up of elongated cells which are arranged vertically and parallel to each other. The oval or round or loosely arranged spongy parenchyma is situated below the palisade cells and extends to the lower epidermis. There are numerous large spaces and air cavities between these cells. Vascular system include vascular bundles, which can be seen in the veins and the midrib. The size of the vascular bundles are dependent on the size of veins. The veins vary in the thickness in the reticulate venation of the dicot leaves. The vascular bundles are surrounded by a layer of thick-walled vascular bundle sheath cell. Look at the figure and find the position of xylem in the vascular bundle. Isobilateral or monocotyledonous leaf. The anatomy of the isobilateral leaf is similar to that of the dorsiventral leaf in many ways. It shows the following characteristic differences. In an isobilateral leaf, the stomata are present on both the surfaces of the epidermis, and the mesophyll is not differentiated into palisade and spongy parenchyma. In grasses, certain adaxial epidermal cells along the veins modify themselves into large, empty, colorless cells. These are called bulliform cells. When the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. When they are flaccid due to water stress, they make the leaves curl inwards to minimize water loss. The parallel venation in the monocot leaves is reflected in the near similar sizes of vascular bundle, except in main veins, as seen in vertical section of the leaves. So in figure 6.5, transverse section of leaf, there is a figure which represents dicot leaf, that is dorsiventral leaf, and B figure represents monocotyledon leaf, which is isobilateral leaf. In dicot or dorsiventral leaf, there is two surface. One is above surface, which is which is formed by adaxial epidermis, and there is another surface or below surface which is formed by the abaxial epidermis. In the dicot leaf, stomata or stoma are present on the abaxial epidermis. On the adaxial epidermis, there are very reduced number or nearly absence of stomata. There is the ground tissue which is differentiated in dicot leaf, and it is well differentiated into palisade mesophyll and the spongy mesophyll. Palisade mesophyll cells are vertical and elongated and parallelly arranged, and spongy mesophills have air cavities. There is a substomatal cavity below the stomata, and there is a presence of bundle sheath which is surrounding the vascular bundles, that is xylem and phloem. Xylem and phloem are more present in midrib or rachis, and the xylem and phloem both help in water and mineral conduction as well as food conduction. In the dicot plant, there is a reticulate venation, that is the veins and venules or veinlets are arranged in a reticulate fashion. In monocot or isobilateral leaf, there is arrangement of xylem and phloem in the middle, or there is a similarity between the monocot and dicot plant. In monocot leaf, there is a parallel venation of vascular bundle, that is parallel venation, that is veins and veinlets. There is no veinlets which form the reticulate venations. There is no network like structures of the venation. There is just the parallel venations. All the veins lie along each other parallelly. And here adaxial and abaxial, both surfaces constitute stomata. Both surfaces are made up of stomata. Both surfaces have stomata for the gaseous transpiration and for the gaseous exchange and transpiration. There is a substomatal cavity below the stomata, and xylem and mesophyll are well differentiated here, but there is no well differentiated ground tissue. There is no palisade mesophyll, there is no spongy mesophyll, there is no air cavities. But yes, there is a water containing cavity is also present, which is considered as lysogenous. So this is all about the dicot and monocot leaf. Summary. Anatomically, a plant is made up of different kind of tissues. The plant tissues are broadly classified into meristematic, that is apical, lateral and intercalary, and permanent, that is simple and complex. Assimilation of food and its storage, transportation of water, minerals and photosynthesis, and mechanical support are the main functions of tissues. There are three types of tissues system: epidermal, ground and vascular. The epidermal tissue system are made up of epidermal cells, stomata and epidermal appendages. The ground tissue system forms the main bulk of the plant. It is divided into three zones: cortex, pericycle and pith. The vascular tissue system is formed by the xylem and phloem. On the basis of presence of cambium, location of xylem and phloem, the vascular bundles are of different type. The vascular bundles form the conducting tissue and translocate water, minerals and food material. Monocotyledon and dicotyledon plants show marked variations in their internal structure. They differ in type, number and location of vascular bundles. The secondary growth occurs in most of the dicotyledon's root and stem. Thank you.